Hi, this is Coach Hikel. I'm back and I'm bringing you a podcast today over cell structure and function. So last unit we talked about the biomolecules that make up cells, like proteins, carbs, lipids, and nucleic acids. And we also talked about enzymes. Well, today we're going to talk about the organelles, or the many organs within cells, and how they help cells function as their own independent living units. So let's get started with the basic history of what cells are and who discovered them and what led us to what we know about cells today. So it wasn't until the late 1600s that scientists were actually able to view living things under microscopes. So up until this time, they could look at things, they had simple magnifying glasses, simple eyeglasses that they could use to see stuff with. But it wasn't until they developed microscopes where they actually able to see things. So the first man who kind of gets the credit for the term cell, his name was Robert Hooke. In 1665, he said that cells appeared to be gel cells. And he made this drawing. He was looking at a cork cell. And he said, these look like cells because if you take this, it kind of looks like a gel cell block. You got these little individual rooms stacked on top of each other. So he actually coined the phrase cell. So Leeuwenhoek, he used a single lens microscope to view pond water and other living things. So he looked through basically a modern day magnifying glass, and he saw these little microorganisms swimming around in water. He didn't really know what they were at the time, but he was the first guy who kind of got credit for saying, hey, there's living things living in this pond water. I just don't really know what they are. So a modern day microscope, we use what's called a compound light microscope. It uses light to view objects. It's good for magnifying things up to about 2,000 times their actual size. An advantage to a light microscope like this is you can view preserved specimens and you can also view living things. So we're going to do both in this unit. We're going to look at some small proteins that swim around in water, and we're going to look at some preserved slides as well. So what are cells? Okay. Basically, cells are the smallest unit of life that can carry out all the functions of life. So one cellular organism, or something that's called unicellular, is actually considered a living thing. Now, humans and other animals are actually considered multicellular. We have more than one cell. We have 25 trillion cells. Okay? So a cell is one thing, carried out all the functions of life. We're very diverse in size and structure. Okay? All cells have a different size. They all have a different structure because their structure and size determines what their function is. And their shape is also related to their function. So if you get really, really big cells, they're not going to function very well. So you want really, really small cells. So again, things can be multicellular or unicellular. So a unicellular cell is made up of only one cell. So this bacterium cell over here is a unicellular organism. It has one cell. That's it. It has everything it needs to carry out life. Most unicellular things are going to be what's called prokaryotic, and we're going to discuss what prokaryotic is in a few more slides. But just know, if it's one cell, it's unicellular. Remember the game Uno, okay? So this cell right here is what's called a multicellular organism. It's made up of many. Each one of these little bitty circles is its own independent cell. They're stacked on top of each other. So this is more complex things like plants and humans are going to be your multicellular organisms. So the modern day cell theory is pretty counterintuitive, but yeah, we got to talk about it. You can say, really, Coach Hikel, I need to know this? Well, there was a lot of work that scientists did before us that actually tells us a lot about what we know today. So it's important to study the past so that we understand where we're going in the future. So the first thing is cells are the basic unit of life. Again, we know that cells are the smallest things that can carry on all the function of life. We know that all organisms, all living organisms, are made up of one or more cells. So a bacterium is considered a living organism. Plants, animals, anything that has those 7 characteristics that we talked about at the beginning of the year, the homeostasis, the reproduction, all those good things that are considered living, a cell can do that. Okay? And we also know that cells can only arise from existing cells. Well, back in the day, there was a man named Francisco Redi, and he actually disproved what was called spontaneous generation. They thought that, these maggots that mysteriously popped out of meat were being hatched by the meat. What they didn't know is that when something died and was starting to decay and decompose, that these flies would land on it to try to get food, and when they were eating the microscopic organisms off of the hamburger meat or the steak or whatever is rotting and decaying, that they would actually lay the maggots into the meat. The maggots would go around their processes and their development, and out these little magical little maggots come crawling out of the meat. Well, we know that's not true. We know that a cell's not going to mysteriously pop out from this computer, and this computer is suddenly going to become a living thing. Okay, so cells can only come from other cells. There's two major classes of cells. There's what's called the prokaryotic cells, which these cells are going to be your smallest, your most simplest cells. Okay, they live in a very broad range of environments. And what sets them apart from other cells is they lack a nucleus and other organelles. So they do not have a nucleus. They're lacking a nucleus. Prokaryotic cells, again, have no nucleus. They also don't have any other organelles. So, like this, they don't have mitochondria. They don't have, the Golgi apparatus, the endoplasmic reticulum. They don't have any of that. Okay? Most of them have a cell wall outside the cell membrane, which helps for structure and support. And many of them use a flagella, which is the tail-like structure, which they use for locomotion. So when you think prokaryotic, think simple, no nucleus, flagella, and an example is going to be a bacterium cell. Another type of cell is eukaryotic. Now the eukaryotic cells are more complex. They're very organized. And unlike prokaryotic cells, they actually possess a true nucleus and all the other organelles we're going to talk about. So they do have a nucleus. These are your plant, these are your animal cells. You are all eukaryotic cells. You got 25 trillion of them approximately. So all your little eukaryotic cells have their own nuclei, they have their own organelles that all function independently, but together to make an organ system, miniature organ system, right? Which creates a cell. So eukaryotic cells. We are all eukaryotic cells. So some features that are common to all cells. All cells are going to have these four common features. All cells are going to have a cell membrane. Now this is not necessarily plant animal. This is prokaryotic and eukaryotic. Okay, we're going to get to the differences in the plant animal cells fairly shortly. So all cells, whether they be prokaryotic or eukaryotic, are going to have a cell membrane. They're going to have what's called cytosol or cell fluid. You might also hear it called cytoplasm. Cyto meaning cell, sol meaning fluid, or plasm meaning fluid. They're all going to have ribosomes. Ribosomes are going to be the protein-making factories within a cell. So they've got to have some way to produce those proteins, those amino acids that we talked about in the previous unit. Remember, because proteins are just long chains of amino acids. Well, that's what these ribosomes do for these cells. And they're also going to have some kind of genetic material, whether it be DNA contained in the nucleus, or circular DNA like some bacterium have, or just DNA that's all clumped together and thrown in the middle of the cell. They're all going to have these four things. I would go ahead and take the time and write these down and know these very, very well, because this will come up and it will appear a lot throughout this unit. So here's a basic picture of the animal cell. We're not going to cover all of these organelles. We're just going to cover the main ones. This is just a basic picture for when you have to go back and label it. You can always pause this podcast. You can go look at stuff, and if you want to go back and say, hey, what's the difference between smooth ER and rough ER? Well, you have a picture of what it looks like, and I'm going to explain to you what's the difference as we go through. So this is an animal cell, and here's a basic picture of a plant cell. Now, the major differences that you can see just by looking at them is a plant cell has a very, very thick cell wall, and it's going to have a very, very large central vacuole. It's also going to have the organelle called a chloroplast. And those are the basic differences that we see just from the beginning. Now, there's other stuff that's different in them, but we're not going to really cover that. So let's just keep it simple for now and say large central vacuole, chloroplast, and cell wall. If there's something else you could think of as you go on there, add that to your list. But those are the three main ones I want you to be concerned with. Okay? So, that's it. So, now we're going to talk about the basic cell structures. It's going to be important for you to write these down because you are going to have an assignment in class dealing with these cell structures. You're going to have to create some analogies. We're going to put them on note cards for you to study for your test. So, it's going to be very important for you to understand the complexity of these cell structures. Now, they're pretty simple. I'm going to try to relate them to stuff you can understand in the modern day world. So if you come up with a better analogy that you want to share with the class, just let me know, and we'll pull you up to the front and say, hey, this is your grand idea. Here you go. We'll give you your 15 minutes of fame. All right, so we're talking about the cell membrane. I don't know why this is all blacked out. I should say cell membrane up here. So cell membrane separates the cell contents from its environment. That's the most important thing. It's made up of 2 layers of what we call phospholipids. Now, thinking back to the previous unit, phospholipids, they're fats. They have phosphorus attached to them, so they're a phospho and a lipid. So it's going to control what enters and leaves the cells. And within this cell, you have what's called these protein molecules right here. Okay, these are going to be, like a channel protein, a carrier protein, some kind of protein. This is a glycoprotein, which is a sugar combined with a protein. So these are going to be the things that are going to be embedded to move stuff in and out. And it's also important to see on this that we have what's called a hydrophobic region, which that's circled hydrophilic. Hydrophobic are going to be these ones in the middle. So these are going to be your hydrophobic, the green. The hydrophilic are going to be the yellow dots. Remember that a phile is something that loves something. So hydrophile, it loves being near the water. If you have a phobia of something, like arachnophobia, you'd be scared of spiders. So these have a hydrophobia. So that means they're scared of water. That's why they're on the inside. So up here, above this cell membrane, we have a water structure. Beneath this layer of the cell membrane, we also have a watery structure. Okay, this would be cytosol. This would just be the stuff floating around outside the cell. So cell membrane controls what enters and leaves the cells. It's what's called semi-permeable. That's going to be an important word to write down. It's actually 2 words. Semi-permeable. So what that means is it lets certain things in but can keep other things out. So for example, if somebody's at our door knocking, they want to sell us something. Those little cute little Girl Scouts that come by, would you like to buy some Girl Scout cookies? Well, most of the time I'm going to say, sure, give me some Girl Scout cookies. So I'm going to open my door or my cell membrane. I'm going to let them inside my house so I can give them money and place my order for my Girl Scout cookies. But let's say I just bought 3 boxes and another girl comes by. Well, we don't want to let that one in, so we don't have to open the door. So we don't open any of our channels. Our channel proteins aren't active. Nothing's going to get in our house. So it controls what enters and leaves the cell. So here's kind of a cross section of everything I was just talking about the transport proteins, the carrier proteins, the glycoproteins. This is how stuff gets in and out of the cell. You got these cell markers which identify the cell type. So if it's not foreign and it's okay, it's going to say, okay, this is a great thing. We're going to let it in. Then you have these receptor proteins. These are what's actually going to bind to it. Then once they bind to the receptor proteins, an enzyme is going to react with it to try to help speed up the reaction. And then you have what's called a transport protein, which is actually going to pull things in and pull things out. And it's going to do it through this channel right here. Okay? It's going to pull things in, push things out, depending on what it needs inside, what it needs outside. Excuse me. So the cell wall. Cell wall is only found in plants. Okay, it's found in some bacterium cells, but for right now, let's just focus on cell wall in plants. It is an inflexible structure that surrounds the plasma membrane. So plant cells don't have muscles and bones and cytoskeletons, so they've got to have a way to stand up. Well, this is what plants have. This helps them stand up. Just like your house has a solid wall around it to keep it from collapsing in on itself. The plants have the same thing. Okay, we talked about how cellulose is a carbohydrate that makes up the cell wall. Different substances and different organisms make up the cell wall. For example, mushrooms or fungi. Their cell walls are made up of chitin. Okay, so that's for another day, but just know that cell walls in plants help them stand up. Cytoplasm. It's also the fluid outside the nucleus contains all the organelles. So whatever left in the cell that's liquidy or fluidy is going to be the cytoplasm. You might hear it called cytosol. Cyto meaning cell. Plasm is fluid. So the fluid of the cell. Many of your chemical reactions actually start within the cytoplasm. So this is the nucleus. These little green dots would be all your other organelles floating around. The light green is going to be your cytoplasm. It's everything else that's not an organelle, not the nucleus, or the cell membrane. So the nucleus, probably one of the most important parts of the cell. It is the brains of the cell, or the control center. It contains all your genetic information. So it also has its own nuclear envelope. So in this picture right here, this is the nuclear envelope. It encapsulates the nucleus. So inside the nucleus, you have a region called the nucleolus. So the nucleolus is going to be this blue area right here. Okay? The nucleolus is the actual part of the nucleus that produces the ribosomes. And a few more slides, I'm going to tell you what happens to those ribosomes once they're actually produced. So the nucleus is like the brains of the cell. If you have a computer, you know, the old-timey computers where you have the monitor and the big CPU tower, the nucleus would be the CPU tower. The monitor doesn't work unless that CPU tower is actually engaged and working. So it has its own nuclear envelope, and the nucleolus is where the ribosomes are actually produced at. So now to ribosomes. Ribosomes are your protein assembly organelles. They make proteins. Ribosomes make proteins. There's no fancy way of putting this. They just make proteins. Okay? Ribosomes connect the amino acids to form proteins. They can be free, floating within the cytoplasm, or they can be attached to what's called the endoplasmic reticulum. And I'm going to show you what the endoplasmic reticulum is coming up pretty quick as well. So, ribosomes, again, are the protein assembly organelles. They make all the proteins for the cells. Okay? Now, ribosomes are made within the nucleolus, but then they make the proteins. Okay? Remember that. Ribosomes make proteins. So, endoplasmic reticulum. Okay? Or ER for short. There's two types. There's a rough ER and there's a smooth ER. So, the smooth ER is the site where lipids of the cell membrane are assembled. Okay? If you have smooth ER, you're assembling lipids. You're assembling them and moving along in kind of a conveyor belt system. Well, if you're rough ER, you're going to be attached directly to the nucleus, directly to the nucleus, because you're going to take those ribosomes that are produced within the nucleolus and you're going to transport them throughout the cell. And you're going to get them out to the cytosol, or the cytoplasm. So if you see an ER with these little peppered little dots on it, those are ribosomes. And that part's going to be directly attached to the nucleus, so that can take the ribosomes, which are made in the nucleolus, out to the cell. So if there are no black dots and it's just smooth, it's called smooth ER. It's making lipids. If it's rough ER, it's transporting. It's transporting proteins. Okay, it's not making the proteins. It's simply transporting the proteins. So again, smooth ER makes lipids. Rough ER transports proteins from the nucleolus, or the nucleus of the cell, to the cytoplasm. The Golgi apparatus. This is kind of like the post office of the cell. It modifies, it sorts, it packages these proteins and transports them from the ER to wherever they need to go. Okay? So what the Golgi does is it takes these, it recognizes, okay, I have this protein. This protein is supposed to go to the mitochondria. It's a mitochondrial ribosome. Okay, we're going to send it there. So got what these called are vesicles. Think of these as the little white postal trucks that deliver the mail. Well, once they leave the main post office, these vesicles take these ERs and other materials. It doesn't necessarily have to be proteins. They take them to wherever they need to go. And that's it. Golgi. It's a post office. Has vesicles, which are the postal trucks. So lysosomes. These are going to be your small organelles. They're going to contain these enzymes. These enzymes are going to break down your lipids, your proteins, and your carbs that are no longer used by the cell. So they're going to get rid of the junk and clutter. So remember lysosomes as being like the Lysol or the trash bin of the cell. They remove the junk and clutter and get rid of all the bad stuff, so that your cells can keep growing and reproducing and keep doing everything that they need to do to be active, and something doesn't go wrong and you start losing brain cells faster than you grow them. Okay? Lysosomes. They're the trash bin. Vacuoles. So animal cells and plant cells both have vacuoles. Okay? We got to have places to store water, salts, and proteins, stuff like that. But plants have a very, very large central vacuole. So it's kind of like the water tower of the cell for a plant. It's going to be the main central thing that you look for that separates a plant cell from an animal cell. If you see a very, very large central vacuole, you know you have a plant cell. Whereas opposed to an animal cell, it's going to have several small vacuoles spread out amongst the entire cell. The mitochondria. Okay, plural is mitochondrion. So these are the organelles that are going to convert the chemical energy that you eat from your food into ATP to power the entire cell. So these are going to be the power plant or the powerhouse of the cell. So they have a multiple layered system here. Before the energy can get to the mitochondria, the reaction occurs in the cytoplasm, which we're going to get to when we talk about cellular respiration. So just remember that the mitochondria is an organelle that's going to convert chemical energy that it gets from food into ATP, which ATP is the energy exchange within a cell. It's adenosine triphosphate. Your body converts everything you eat eventually into ATP so that you can power your cells. You have lots of cells that does this many times a day. So you're going to have a lot of these mitochondria. So if you think about the different cells in your body, you have like skin cells and you have muscle cells. So which one of these cells would you expect to find more mitochondria? A skin cell, which doesn't do a lot, or a muscle cell, which is constantly reacting to move your muscles, to move you, to contract and constrict and hold you up and do all this supporting? Your muscle cells are going to have more mitochondria than a skin cell or a brain cell. Okay? They need it because they're using more energy. Chloroplasts. Chloroplasts are found only in plants. Okay? Found only in plants. These are the guys that are going to capture the sunlight and convert it into a chemical energy through a process known as photosynthesis. Okay? So wouldn't it be great to walk outside and go, ooh, I'm fully energized. Our chloroplasts are working. Well, plants get to do that. They get energy from the sun through a process called photo, which means light, synthesis, which is making. So they're making energy from light. Okay? Chloroplasts are found only in plant cells. So, common misconception is that animal cells have mitochondria and plant cells have chloroplasts. Most people don't realize that plant cells also have mitochondria. They don't have as many as animal cells, but they are going to have some chloroplast. And we're going to have another podcast that's going to talk about endosymbiosis, which you're going to see the development of mitochondria from chloroplast and all that good jazz coming up later. Okay? So within the chloroplast, there's several structures that you do need to know. You do need to know that these granum are what's called stacks of thylakoids. We have a thylakoid disc, okay? These individual little circles are going to be your thylakoid disc. When they stack together, they're going to form what's called a grana, okay? Just put in those two words in your head right now. We're going to get to them when we start talking about photosynthesis. So the thylakoid discs are actually going to capture all the sunlight, and a stack of thylakoid discs is called a grana or granum, which is the plural form of it. Cytoskeleton. Cytoskeleton, think of this as the bones or the muscles of the cell. It's going to help the cell maintain its shape. Now, would you expect to find this in an animal cell or a plant cell? Think back. I told you that plant cells have cell walls to help them support and maintain their shape. So this is mainly in animal cells because it also helps us move our cells. It helps us keep the flexible membrane, but still keep it rigid enough to where it can support our body weight and do everything that it needs to do. So, cytoskeleton. So, that's it for today, guys. I hope you enjoyed that a little bit more than the last ones. I have a little more energy this one. I was under the weather for the last one I made. So, as always, if you have any questions, please feel free to email me or ask me in class. Do whatever you need to do to get this material. We will be making some note cards over all the organelles. We will be looking at some different types of cells under microscope slides. So, this is Coach Hikel, and as always, have a great day. Bye.